Okay, so we've been talking about bacteriophages for most of this. We're going to focus on animal viruses, and then we'll talk about other infectious agents really quickly at the end of the lecture. So we will continue on. Phages versus animal virus. And phage, if I haven't explained it already, is just a shortening of the word bacteriophage. They both mean the same thing. So viruses that infect animal cells are a bit different than phages due to the structural differences between bacteria and animal cells. Recall that bacteria cells have rigid cell walls. They do not permit entry of viruses. Bacteriophages inject their genome through the cell wall, but they do not pass through. So we've been talking about that at length in this lecture. But animal cells, however, do not have a cell wall, but they're surrounded only by a phospholipid bilayer. So remember that from your high school biology classes. Animal cells also express surface receptors on their membranes, like we mentioned. Bacteria cells do not have receptors because they have a cell wall. So because of this, many animal viruses take advantage of these receptors and enter the cell via endocytosis. Alternatively, some viruses surrounded by a membranous envelope can automatically fuse with the membrane to release their capsids into the cytoplasm. And I put a picture at the bottom so you can get a sort of visualization of how a bacteria cell and an animal cell are a little bit different. The bacteria cell to the left, you could see they have those little cilia sticking out and then those two flagella at the end. And that rigid yellow cell wall protects the cell membrane and the cytoplasm from outside stresses and specifically certain types of viruses as well. And then you look over to the right, the animal cell over there does not have a cell wall. It's only surrounded by a plasma membrane. And if you actually look to the bottom right, you can sort of see a sort of budding inward of that membrane. And that's really what it would look like if the virus were taking advantage of endocytosis or fusion. So it would sort of bud out. You could see that little pinching of the membrane. The pinching area is going to come together and we're going to have a little vesicle that is inside of the animal cell. So that's how viruses gain entry into animal cells. So animal viruses may also enter lytic and lysogenic cycles after infecting a host cell, very similarly to how bacteriophages do it once they're inside of that bacteria cell. When a viral genome is inserted into the host genome during the lysogenic cycle of an animal cell, it is known as a provirus. So I used the word provirus a couple of minutes ago. When we're talking about a bacteriophage in bacteria, entering the lysogenic cycle and they're inserting that genetic information into the host cell's genome, that specific part is called a prophage. When we're talking about animals and animal cells entering lysogenic cycles, the genetic information is called a provirus. So it's very similar. It's the same concept. We just use different words because it's an animal virus and it's not a bacteriophage. So we say provirus. So those are pretty much exactly the same, the lytic and lysogenic cycle. Animal cells, though, can actually enter a third cycle, which is known as the productive cycle. So the productive cycle is sort of like, it's sort of like a slightly altered lytic cycle. It's almost identical, except at the very end. So instead of lysing the cell, the cell remains intact. So the viral cell inside increases, but because there's no cell wall, these viruses, remember, they came in via fusion of the membrane, or they came in via receptor-mediated endocytosis. They can actually exit the cell in the same way. So if they have that envelope, it's very easy for them. Not even before they have the envelope, it's easy for them to bud out of the membrane. And when they bud out of the membrane, that's what actually forms their envelope. So if you look to the right over here, this describes everything. So over here at 1, you could see the actual endocytosis when it's going in. You can take a look at this on your own if you'd like, but when we come up here to 6, you can actually see it budding outward. So you can see how this membrane actually starts to form around that protein capsid, and it's eventually going to push outward, pinch off, and it's going to be surrounded by that own plasma membrane that was made up of the original cell membrane. And that animal cell, it's designed to do that. So it stays intact. It doesn't lyse like a bacteria would at the end of a lytic cycle. And that's pretty advantageous because the host cell remains intact and it's actively producing new viruses they are released. So remember, with the lytic cycle, once you have 100 or so bacteriophages, that cell is going to burst and it's not going to be able to produce anything else. But with this productive cycle, and we call it productive because it is productive compared to the lytic cycle, we can just keep on using this animal cell for as long as we possibly can without lysing it. Okay, a little bit about HIV and retroviruses. HIV, which is the human immunodeficiency virus, affects the immune system of humans. And I just put this on here really quickly because when we talk about viruses on the MCAT, they very much like to talk about HIV, hepatitis, but HIV very specifically, I'll see a lot of passages regarding. So it's very good to have a pretty decent background in this, even though a lot of times we will see it in context of passages on the MCAT. It's a good idea to have background information about it. So if you don't already know, HIV affects the human immune system. It expresses a protein that can interact with the CD4 receptor on human T cells. So remember, we talked about that receptor-mediated endocytosis. Those CD4 receptors are used in everyday immune functioning, and the HIV virus has found a way to sort of hijack this system and gain entry into the human T cells, where it destroys them and multiplies. So as the viral count increases, the white blood cell of the individual is going to decrease. The individual becomes susceptible to other infections, and these infections severely debilitate, and they eventually kill the individual if the individual is not being treated. I mean, nowadays we have a lot of different treatments for HIV. We have antiretrovirals. We have a bunch of different treatments. There are constantly like new studies being performed on different types of treatment, different cures. So it's really interesting. If it's something you're interested in, it's definitely worth looking up. There's some really cool research out there on HIV prevention and curing, actually. So HIV is classified as a retrovirus, and a retrovirus is a virus with an RNA genome that undergoes reverse transcription to integrate viral DNA into the host cell. So that's sort of part of the lysogenic cycle. Remember, with a bacteriophage, it's a DNA virus. So it injects its DNA directly into the cell, and then that can be automatically integrated right into that cell's genome, right into the bacteria cell's genome. But if we have HIV, which has an RNA genome, and we infect a human cell, we can't integrate RNA into a DNA genome. It just doesn't work like that. So these retroviruses have come up with a way to change their RNA into DNA and then insert it into the host genome. And what they do is they encode for a protein that we call reverse transcriptase, which is also known as RNA-dependent DNA polymerase. So what that does is it takes RNA as a template and it creates DNA, which obviously is opposite from the central dogma. It always goes DNA, RNA, not always, but mostly DNA, RNA, protein. So this reverse transcriptase allows them to sort of defy that central dogma, create DNA from their own genetic information and integrate it into the host cell as a provirus. And that's one of the super tricky things about HIV. So we have a bunch of pre-exposure prophylactic medicines, and what that does, they're basically reverse transcriptase inhibitors. So if you're taking this constantly before HIV infection and you come in contact with the HIV virus, the reverse transcriptase is not going to work initially enough to create enough DNA to integrate it into the host genome. The problem is that only works before the HIV is able to create DNA and integrate it. So once that DNA is integrated into cells, it is much, much, much more difficult to get rid of those genes that are encoding for this HIV infection. So that is one of the hot topics of research too, is how is it possible to target cells that have this HIV provirus in it? How do we get rid of it? Because like I said, once it's in there, it's very difficult for the cells. It's very difficult for your immune system to even notice anything is wrong until these actual that part of the genome is actually being expressed and creating that virus. If it's not creating the virus, it's very difficult to see. So positive and negative RNA strand viruses. Viruses with single-stranded RNA genomes can either be positive or negative strand. Positive strand RNA viruses carry a genome that act directly as mRNA in the host cell. So the DNA, the RNA that they carry can immediately be transcribed. It can go into the cell, and the ribosomes of that host cell are going to read this RNA, and they are going to immediately start transcribing those proteins. For this reason, we refer to these genomes as infective. So positive sense RNA must encode RNA-dependent RNA polymerase because animal cells do not create RNA from other RNA templates. Again, remember the central dogma. So retroviruses, we have RNA-dependent DNA polymerase because it's using RNA to create DNA. And now for these sort of viruses, we're using RNA-dependent RNA polymerase. We're creating RNA from other RNA. And again, human cells don't do that. We create RNA from DNA. So we don't encode for this gene. So these viruses have to, if they're that positive strand DNA. So RNA-dependent RNA polymerase must first create a complementary strand to use as a template before replicating the infective RNA from its genome. Because remember how polymerization works, it's all complementary. So if we create a complementary strand from that positive strand RNA, and we try to make a protein from it, it's gonna code for, a useless polypeptide, basically. It's not gonna do anything. So that's why we have to create that complementary strand from this positive sense, and then those complementary strands can create more of those positive strand RNAs, and then that's how these viruses create RNA. So viruses that cause the common cold are examples of RNA viruses, of positive strand RNA viruses, excuse me. So on the other hand, we have negative strand RNA viruses that carry the template strands to create mRNA. So the positive strands, that RNA genome can immediately be used as mRNA in the host cell. The negative strand RNA viruses only have the complement, that template strand. So they need to also use RNA-dependent RNA polymerase, but they actually have to carry that protein. If they only encode for that protein, there is no possible way that it can be created when it enters the cell, because all it has is that complementary negative strand RNA. So if we have no proteins and just that RNA, there's no possible way that we can make that complementary strand, because our host cells, our human cells, do not code for RNA-dependent RNA polymerase. So these negative strand RNA viruses have to have this protein already created in order to be able to function and create more. Right, because the genome can't be translated to usable polypeptides, they have to carry the RNA-dependent RNA polymerase. And examples of these RNA viruses are rabies, measles, and influenza. So if you want to take a minute as well, you can look to the right. There's a little bit more information about positive sense RNA viruses and those negative sense RNA viruses.